right, so this talk might have been a little better placed earlier in the day when we were talking about healthcare, but this is sort of the last of the healthcare talks. And so I'll maybe be able to, thank you again for coming. I'll be able to maybe help fill in some of the terminology and things that were presented earlier. So I'm going to talk about what I'm calling an adversarial view of biological communications. And so those of you who know my role and history at WINLAB, I was really focused on security and I guess even electronic warfare. And after a while, I started looking into other topics, because as you get older, you want to go do different things. And I started looking at biological communication. So this is a different form of cellular communications. This is communication between cells in our bodies and stuff. So I'm going to start off with a little cartoon, and this is as good as my cartoons get. So over here we have a little bacteria. So he even says, hi, I'm a bacteria. All right. You might ask, how do we know he's a bacteria? So what comes along is your first line, your innate immune system. All right. So a macrophage is a good representation of it. Its job basically is to go look for things that don't really fit in your body. All right. We'll talk a little bit about how we recognize those things. And through some friend or foe detection technique, it says, hey, this is not a friend, all right? So I'm going to go eat you, all right? And it basically goes through phagocytosis. It eats it, chomp, chomp, chomp. All right, and then it sends out signals to other cells in your body. All right, now how it does it through a variety of different techniques, all right? And in the process of doing so, all right, it calls in other parts of your immune system. It calls in dendritic cells. These dendritic cells, their purpose is basically to take antigens that were on the surface of that bacteria and basically present them to other parts of your white blood cells, other types of white blood cells, and call them in. And so what he does, all right, back when my son was interested in Avengers, he says, time to bring in the T cells. T cells assemble. And so all of your T-cell lymphocytes come in and they basically start really helping you go through your adaptive immune response. So these biological cells, they communicate by releasing different types of chemicals. These chemicals typically are proteins or hormones, all right? And so you might have heard the term cytokinin earlier this morning. Cytokinin is basically a protein that's being released, and it causes a variety of your body's responses. It's partially responsible for your when you, I guess, swell or go through hot flashes and stuff, it's your body really trying to shake, say, a cold or something. All right, other things, hormones like growth factors and so on get released. Now, interestingly, a lot of our modern diseases that we are aware of, all right, are due to the fact that these signals that are going between different parts of our organisms have broken down in one way or another, all right? Either we're not getting enough of these signals, all right, or the signals are too much. And so a lot of medicine is about trying to adjust those communications, all right? And in particular, interestingly, cancers are often described or envisioned as processes that hijack our normal communications. So for example, a lot of cancers tear down the ability to receive the time to die signal, all right, for cells. All right, so you guys being communications people, you'd like to understand and find a connection between that little macrophage and what we do. So we're all familiar with point-to-point communication. So basically I picked up a wire, all right, communicating from one end to the other. All right, it's direct contact for the communication. And in the wireless world, we're familiar with broadcast and multicast. The medium sends stuff out in all directions. All right. In the biological world, we have something similar. All right. Instead of transmitting the waves, we emit signals. All right. These signals are in the form of proteins or actual direct contact. So over here on the left, we have what's an example of a juxtacrine signaling system. All right. This is actually neuron signaling, so one neuron connects to the next neuron. They're basically touching, all right? Next over in the middle is the paracrine signaling. All right, paracrine is basically a local but still broadcast, so you release these chemicals, all right, that go out and they hit the target cells, and the target cells tend to be pretty local to the originating signaling cell. And last is your endocrine system. That is basically, think of it as your hormone system, all right? And so, you know, the actual production of hormones goes through your bloodstream, circulates, and arrives at the target cell. And as an example here, we have what's called a tropic hormone. All right, a tropic hormone is a hormone whose responsibility is to regulate other hormones. And so the growth hormone regulating hormone, that's the GHRH, its job is basically to, it's released by your hypothalamus. Its job is to go to the pituitary gland, all right, and tell the growth hormone producing cells in your pituitary gland to either produce or to stop producing. And so it then gets received by a cyclic AMP receptor, all right, that's on the surface of the cell, all right, so it does some binding, all right, and then that causes the release of this growth hormone. So this notion of that receptor, the one that I drew up here, all right, and actually I should say diagram from nice talk from University of Washington, so I didn't draw it. So the notion of this receptor is key to modern medicine. Right? If you go back to the 1800s, a lot of the way medicine was treated was just your blood, all right, you know, leeches and really bizarre, you know, fictional ways of dealing with things. It was in the late 1800s where they began to have this idea that, well, maybe there's actual chemicals that are going on in our body and they bind to things. Much later, in the early 1900s, is when the formal theory of receptors was developed. And so here we have a biological cell with a cell membrane, all right? We've got receptor molecules on the membrane of the cell. And then we have, up here in the top layer, these are pharmaceutical agents, aka drugs, or they're just other chemicals that your body naturally produces, such as hormones, all right? And what happens is when the right-shaped item gets to the right-shaped receptor, all right, it causes, for example, a release of something, another signal, another chemical inside of that cell. All right. What's happening is that binding is causing this protein, which had it attached, to change its conformation, and it releases the second signal. All right. So, taking some diagrams from a standard textbook, what's interesting is that it's not quite as simple as that diagram that I showed you in the previous one, but that this receptor then initiates a long process, a process we call transduction. And for those of us who are communications people, it's a network, all right? It's not a simple, oh, a signal gets released and then it goes on to the nucleus, all right? That little process here looks like this, all right? And these are all the different chemicals that then get fed to another chemical that then leads to this really large interaction network, all right? And ultimately, it gets down to the bottom right here, which is in the nucleus, where perhaps some type of genetic modification is being made, all right? And as we start looking at this receptor and how it leads to different pathways that are being actuated, all right, there are many different ways that it could happen, right? One receptor could activate two separate pathways inside of a cell, all right, or two separate receptors then together are needed sort of as a combination logic, or an AND, in order to then initiate a pathway, or bizarre complex versions of these. And what that means for medicine is that you have many different ways in which you can have benefits, so good therapeutic responses to treatments, all right? So a drug gives you something good, but unfortunately often it will also give you a toxic side effect, all right? And there are many different ways that these benefits and toxicities begin to manifest themselves when you look at something like that diagram there. All right, so as I was mentioning, modern medicine is about understanding how these chemicals affect our signaling pathways, all right? And the human body, unlike, well, in a way, a human body is a little more fancy than wireless. Wireless is wireless, right? It's just div, grad, curl, and, you know, E and M. But for biological systems, they have a limited set of signaling compounds that they use. The amount of proteins that are being used and reused for multiple purposes inside our body is still, although larger than just div grad curl, is still quite limited. And so here in this diagram are some of the different functions of our body that are affected by adrenoreceptors. All right, so when you take something like epinephrine, all right, it's good for bronchial dilation. So we had a talk earlier about COPD and breathing. So you would take something like epinephrine, all right, which binds to your adrenoreceptor. But unfortunately, the side effect of that is, well, it also changes your heart rate, or it could lessen the actual force that your heart is pumping blood through your system. All right, we reuse inside of our body the same chemicals for many different purposes, and that's one of the hard parts about medicine. All right, a couple quick definitions as I move on. Agonists are basically chemicals whose job it is to bind to something in order to produce an effect, and antagonists are chemicals whose job it is to block something to prevent an effect. And all of medicine, we wish, looked like this, all right? This is what's called a dose-response curve. So here on the x-axis is the concentration, all right? And for our community, we can think of that as signal-to-noise ratio, all right? As I increase the signal-to-noise ratio, all right, I get more bits through, right? Or I, more effectively communicate. This is essentially a bit error curve, or flipped upside down, for drug communication. All right, so here we have increased concentration, and then the effective response of one drug versus another. All right. What is different, though, is unlike the Shannon log of 1 plus SNR type shape, we tend to get sigmoids in I guess pharmacokinetics or pharmacodynamics. All right, and then we have this notion then of different modulation schemes are better at reducing bit error rate than others. Similarly, we might prefer a curve over here to the left, which says I get more effective response quicker than if I went further to the right. So I increase the potency of my drug by moving to the left. And likewise, all right, I might be able to only activate half of the receptors on a cell. All right, so I get half receptor binding, whereas a different drug might be able to activate all of them. All right. And then the bad news. The bad news is you have these curves for what's good for you. You also have these curves for what's bad for you. All right, you take a medicine, it's helping you, but unfortunately you also begin to kick in the harmful stuff. And so then that says, all right, what can we do with this basic medicine 101 and relate it to communications? Again, communications is about building systems with trade-offs, all right? And one of the first things we can do is look at trying to trade off between these harmful and beneficial side effects, all right? Another thing we can do is look at this like a channel allocation problem where I'm putting power into different drugs instead of different channels, all right? And we can then begin to look at other insights that we can make motivated by communications. So the first problem I want to briefly mention is then looking at the trade-off between what is a therapeutic response. All right, so you're taking a medicine to deal with an actual disease you might have, but unfortunately you're getting side effects. All right, and you can formulate this through a standard Nash bargaining communication trade-off type formulation. All right, since we know ideally what these functions look like, and they're represented through basically sigmoid functions. All right, we can try to bargain between a benefit and a harm. All right, and it's really, there are nice ways to formulate this. Again, this was not a tool, though, that was used by the pharmaceutical industry. All right, but by looking at their problem and then recasting it as a communication problem, all right, what my group and I were able to do is to formulate this basically as a mathematical problem for biology. It's a trade-off problem. And it was really, really very nice for us in that we got a paper accepted for a brand new area that we were starting at, and they had never seen this tool. Now, one thing you quickly get from here is that as you start increasing, these functions ramp up pretty fast. All right, most drug response curves look like that. And unfortunately, all right, the bad curve also ramps up pretty fast. And so what can you do? Well, one thing you can do is try to use multiple drugs. All right? Just like one thing you can do with OFDM communications is use different subcarriers that give you better, I guess, fading characteristics than the bad subcarrier. All right? You can do that also with medicines. All right? And so as a motivating example, and I'm borrowing this from my grad student, when you have a headache or a migraine, all right, you might take a variety of different compounds. All right, so aspirin or Tylenol, all right, or go get another coffee, okay? Well, interestingly, there's a combination medicine out there that is a small amount of each of these that has been shown to be more effective at total lower dosage than the individual compounds separately, all right? It's a synergistic effect. And that's sort of what we want. We want to find teamwork, all right? In all of engineering, we want to find situations where you put two things together and you get more than if you had the two things separately. And so how do we do that? Well, one, we begin to look at these curves and look for points of diminishing return. And we say, okay, if we go too far this way, I'm having to put a lot more energy in order to get a little bit of return. All right? And guess what? As I put a lot more energy to get a little bit more return, I didn't see if the animation does it. Yes. I've also begun to kick up my adverse, my toxic response also. So if I need to get way out here, well, I already now suddenly jumped up in my toxic response, you know, the one I plotted with red. So we ideally would like to stay back over here, where we ramped up quickly before we got to this diminishing return. Well, we can't do that with one medicine often, but we can do that with multiple. We can use multiple drugs, each operating down here in this regime, all right, to effectively add together. All right, and so I'll move past this. So these multiple drugs can have a lot of benefits to each other, and there are numerous examples out there. I just mentioned migraines and headaches, all right, HIV treatment and cancer treatments. Most treatments ultimately rely on trying to take advantage of multiple drugs to deal with the problem as opposed to a single. All right, it helps reduce toxic side effects as well as some other things that I'm going to mention shortly. So cancer and many other diseases often involve multiple problems simultaneously. And I'll show an example of a network to sort of illustrate this in a little bit. All right, but the idea is if we could hit a sickness like cancer or my headache on multiple fronts, then we are more effective than hitting it on a single front at any time. And this happens for a variety of reasons. One, in the signaling pathways like I showed earlier with lots of interconnected networks, all right, we can hit with multiple drugs different places in that network. All right, and hitting it in multiple places tends to be more effective than just hitting it one place alone. And then the other reason, all right, is to recognize that we as humans, all right, are quite diverse. All right, my response to one medicine might be different than his response. All right, or in the case of cancer, all right, a tumor is not one single thing usually. It is many different types of subpopulations with different responsivenesses. All right, and so by hitting it with a single drug, I might kill one subpopulation, but I didn't kill the other subpopulation. And that is often the cause of what's called metastasis, right? When cancer metastasizes, it's because you killed one particular subpopulation, but you let the other subpopulation hide under the radar and it began to grow later. All right. The trick, though, is to find ways to find these multiple combination drugs. And it's not as easy as just go to the lab. Lab, as we were mentioning earlier, all right, running these trials is extremely expensive for the pharmaceutical world. And so we like to have better mathematical models. And what's happening now is that this AI and machine learning and all that is not just hitting the wireless world, but it's hitting the medical world. All right, and they're looking at these type of signaling networks, trying to find combination drugs and hypothesize whether they'll be productive or not. And then we're trying to, what my group's trying to do is trying to put some mathematical models. And watching my time, I'm going to skip through some of this stuff here quickly. All right, so there's this notion of trying to quantify when two drugs work together, all right, are they synergistic or not synergistic? Are they working together more effectively than if they were hurting each other? So here is a curve of drug 1 versus drug 2, their dosage level, if you will. And this line 1 minus x, if you will, all right, represents sort of the line where if I fall below it, I'm having synergism. I'm having more benefit combining drug 1 with 2 than if I was just to use them separately. And antagonism is above the curve. All right? And we are looking for things that fall down here. And unfortunately, it's not an easy thing. And there are a lot of models, and I'll move into that in a second. A lot of models, though, that try to determine, am I going to have synergism or not? And in order to do that, we need to understand what are all of our tasks and objectives in trying to find medicines or trying to affect the control of biological communications. All right? And it's basically an optimization problem. I put it together this way so it looks like the standard type of programming problem, linear programming that we in the communication resource allocation world like. We want to maximize something while minimizing something. All right? So what do we want to do? We want to maximize like the response, all right? The amount of patients that successfully respond to treatment, while we're minimizing, say, the adverse effects, or keeping all dosage below a certain level, or reducing drug resistance. That's another problem that shows up. And at the end of the day, we find some optimized dosage schedule. And as I was mentioning, and this is where I'm going to fast forward through stuff, there are a lot of mathematical models that were developed, say, 20, 30 years ago. And ultimately, these ended up being the equivalent of what we would call a black box model. All right? And what's happened is there's been a shift in the last 10 years or so to trying to look at the underlying network of a disease, how the disease manifests itself through different pathways being altered and affected. All right? And if we start looking at the relationships between, say, good pathways and bad pathways, what can happen is we can find combinations through the topology of their interactions where I could be more effective by combining two drugs at a particular combination zone than if I was to do one separate from the other. All right. Now, I have about 5 minutes, so I'm going to wrap up here with talking about the network perspective. The unfortunate thing is that this network that I showed right here is really it's a toy example. It's really very small. All right? And this is a little bit closer to what a biological network looks like, but even this is a simplified version. All right? This one here is epidermal growth factor signaling. This one here, all right, is toll-like receptors. All right, and guess what? This is also, unfortunately, highly simplified. All right? And if you take each of these lines, what you begin to find out is each of these lines is like its own 20-dimensional stoichiometric equation with reaction rates and phosphorylations and so on. All right? But it's really complicated. But what people are doing, like we do in network design for communications, is we look at those type of diagrams and we try to find patterns that show up over and over. These are called network motifs. And these are the 21 network motifs that lead to synergism for 3-node subnetworks. All right, and what that means is we could go back to diagrams like this, try to find these 3-node pathways, all right, that look kind of like this, all right, and understand them and maybe try to target those or attack those. And so that's part of the program that my group is working on, is trying to understand, if we take something like these feed-forward diagrams, how can we target and disrupt a feed-forward diagram in a biological circuit? And so this is really similar to trying to target a 3-node communication network, where I'm introducing jamming interference here, 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 by trying to slow down the reaction rate of those equations. Likewise, this is what's really kind of cool. All right, it's not all communications; it's also control theory. All right, so all the control theorists, all right, might be able to come back into vogue now. All right, we find a lot of feedback mechanisms inside of biological networks. All right, so your metabolic network says, "Hey, you're full. Time to slow down on eating." Right. So the nice thing, if you've had control theory, is like feedback is this really amazing thing. All right, you don't have to actually really understand your problem. You could build a negative feedback loop, not have a perfect physical model, and it will still do its job. All right, and that's one of the great things about feedback loops, is you get decreased sensitivity to variation parameters. I don't know the exact parameters inside my loop, but I'm still quite stable. All right, I can reject noise. All right, and I end up converging to less error than if I had otherwise had a model. All right. Unfortunately, if this goes wrong, it goes really wrong, and that is one of the causes of our autoimmune diseases like lupus and rheumatoid arthritis. On the other side, positive. Oh, actually, let me mention, why do we care about this? Well, it also has an implication for drug discovery. All right, if you're trying to target something that's inside the loop, don't do it, because that's stable. All right, the loop is going to correct for any perturbations you were able with your drug to introduce to the process plant. Guess what? It's going to correct for itself because of the feedback. That's what feedback does. It corrects for imperfections. So the end lesson that came from all this network pharmacology was we should try to target processes that are outside of these loops. Don't mess with feedback loops. I'll skip feed-forward loops. All right. So how do we do this? Well, there are a variety of different ways, and this is getting to the end of my story. All right. One thing we could do, we could try to take a network, understand its connections and the strength of those connections to find the most strategic nodes or paths to disrupt. And again, that's like a standard jamming problem for communication systems, and one that we and others have looked at using graph connectivity and fiddler based metrics to find where to strategically puncture a network. Another thing is to look at this as a network flow problem. All right, this is like bringing back old operations research literature. If there's an overall flow to this diagram, all right, how can I strategically place things to most disrupt that flow of that diagram? And that's a problem that shows up a lot in sort of old military operations research literature. How do I disrupt my enemy's ability to resource itself? And then lastly is to try to apply techniques from game theory and evolutionary game theory to understand the us versus them dynamics. And so I will wrap up here by saying that there's a lot of really cool stories out there. And in my past couple of years when I started shifting my research to this area, I've discovered all sorts of new things that are just, it's really quite interesting, the dynamics that biological systems have evolved towards. All right, so for example, I'll just point this one out. Quorum sensing is a way by which bacteria sense that other bacteria are near them. All right, and if there are too many bacteria, they're going to compete for their limited amount of resources. All right, and so what bacteria do is they adjust the rate at which they replicate themselves so that they won't starve themselves out. All right, well, enter bacteriophages. Those are viruses that come over and hijack bacteria. Well, what they end up doing is they hijack also the bacterial quorum sensing mechanisms to say, okay, I'm going to use the bacteria to replicate myself inside this bacteria. But if I choose to destroy the bacteria and spread myself out, which is what my function in life is, if I do that and there are not enough bacteria around, I will basically waste my energy. So they've stolen the sensing mechanism from bacteria in order to make certain that they survive. It's really kind of cool. There's all sorts of examples of communication going on. And so to wrap up, though, here's the hard part. The part that I've learned, which is a lot of this research, a lot of what's out there is unfortunately not very mathematical. It's very laboratory oriented, all right? You know, that sigmoidal response, that's a theoretical curve, but when you see it in a real like clinical data, all right, it's like 5, 6 data points and then they fit something to it. A lot of stuff is just not enough data. All right, so we need better data, and that was the first talk, all right? Well, I wouldn't say the first talk, one of the early talks by the guys talking about biosensors. In order to get biomarkers, which are real ways to collect the data we need to understand those networks, we need more advancements in biosensors. And then ultimately, that will allow us to do this quantitative analysis that us as engineers love to do. So I'll wrap up there.